Now, when proteins are synthesized, they function for a certain period of time. They're going to do their job, but eventually they're going to get degraded. Now, how does a protein get degraded? There's different ways in which a protein can get degraded, and I'm going to show you one of the major ways which you're expected to know. There's a protein called ubiquitin. Now, just like its name suggests, it's a ubiquitous protein, so it's found just about in every cell. Ubiquitin, when it gets attached to a protein, it's going to tell that protein, I need to be degraded. So it's going to send the protein to a part of the cell called a proteasome. And as a matter of fact, different proteins, as they're made, they might get misfolded. And so it's never going to be a good protein. There are mechanisms in the cell that we don't have to go into by which these misfolded proteins are recognized. Ubiquitin is then attached to the protein through a process that we call ubiquitination. So ubiquitination attaches ubiquitin to these misfolded proteins, and it takes the proteins to this proteasome complex. Inside the proteasome complex, the protein is going to be degraded to small peptide fragments, and the small peptide fragments are going to probably be further degraded. There's a couple of key points I want to mention here. First of all, there's a class of proteins called chaperones. Chaperones. When you hear the term chaperones, they aren't people that make sure you're not kissing at the dance. Chaperones are proteins that help hold a protein in a proper combination. They're helping hold the protein in the properly folded conformation. If this mechanism fails, then the ubiquitin can bind to the misfolded proteins and bring them to the proteasomes. The other thing I wanted to mention is that this is what we see when we look at antigen processing in immunologic cells. So, in a cell that's going to process an antigen, chop it up, and then display it on the surface of the cell in the context of either MHC1 or MHC2, the major histocompatibility proteins, well, this is how it happens. It's going to chop up the protein into peptide fragments. These peptide fragments are going to be associated with either MHC1 or MHC2, depending upon the cell type. Okay? And then those will be displayed with perhaps a T cell receptor or some sort of B cell receptor that can then get further bound. You'll get that reviewed again for you in immunology. Let's now talk about what happens to a protein after it's been synthesized on the ribosome. Most proteins, before they're considered fully functional, have to be modified in some way. So they're either going to have to be hydroxylated, glycosylated, sulfated, phosphorylated. They're going to be modified in a way that allows them to be sent to the proper part of the cell or even secreted from the cell. For example, if a protein's meant to be membrane bound, it has to be modified in a special way. If a protein's going to be sent to the lysosome to function, it's going to have to be modified a special way. If a protein's going to be secreted from the cell, it's going to have to be modified in a separate way. How does that happen? Well, typically it happens within the confines of the endoplasmic reticulum. I think of the endoplasmic reticulum like the dressing room of the cell. It's where proteins go to get all gussied up and get all fancy and put on modifications before they go out to party, right? So that's the endoplasmic reticulum of the cell. How does a protein do this? Let's take a look. Now, initially, translation begins within the cytoplasm. But immediately after translation begins, the ribosome, for most proteins, is going to go and it's going to bind to an endoplasmic reticulum. There's a special way in this has to happen, and that's where the board exam likes to test you. There's something called an N-terminal hydrophobic signal sequence. I want you to remember that. N-terminal hydrophobic signal sequence. At the 5' end of every single protein that's destined to be modified in the endoplasmic reticulum, there's a hydrophobic sequence of amino acids. Now, remember that there's certain amino acids that are hydrophilic, hydrophobic. The hydrophobic ones were like proline, alanine, valine, leucine, isoleucine, glycine, tryptophan, tyrosine, phenylalanine. These are hydrophobic amino acids, and these are going to tend to be clustered at the 5 prime end of the gene, but at the N terminus of the protein. So the 5 prime end of the gene would correspond to, of the coding sequence, which would correspond to the N terminus of the protein. Now, these hydrophobic amino acids are going to bind to something called the signal recognition particle. The signal recognition particle will thus bind to the hydrophobic signal sequence and guide the protein ribosome mRNA complex to the endoplasmic reticulum. Now, shown here, the protein that's now been bound to the signal recognition particle will be fed into or extruded into the lumen of the endoplasmic reticulum. The endoplasmic reticulum has these hollow tubes, and the protein will be fed into that hollow tube. Immediately, what usually happens is that a signal peptidase will cleave off the signal sequence. So the N terminus of most proteins has hydrophobic amino acids. This is important for getting the protein to the endoplasmic reticulum, but right after the protein gets in, the signal peptidase cuts off those N hydrophobic amino acids. Now, translation is going to continue on the endoplasmic reticulum. This is why, if you take an electron micrograph of a cell, you see that the endoplasmic reticulum has all these dark, electron-dense dots sprinkled, like poppy seeds all over it. What are those dots? Those are ribosomes. As a matter of fact, there's something called polyribosomes. A couple pages back in your syllabus, you'll notice we talked about polyribosomes. Polyribosomes are mRNAs that are bound by multiple ribosomes. So many ribosomes can be bound to one single messenger RNA molecule, and all of these are individually making proteins that get fed into the endoplasmic reticulum. That's a polysome or a polyribosome. Now, once within the ER, this is where the proteins are going to get all decorated up. They're going to put on earrings, and they're going to put on jewelry and lipstick and perfume, right? They're going to get all ready before they go out into the big world. Well, what am I talking about? No, they're not going to put on earrings and lipstick and perfume. They're going to put on hydroxyl groups, phosphate groups, sulfhydryl groups. They're going to get things added to them that allow them to function later on. Okay, so within the ER, the particular mechanism we're going to focus on for now is glycosylation. Glycosylation happens within the ER, and what you see here are these little sugars that get added to specific amino acids. Now, within the endoplasmic reticulum is where N-glycosylation works. It's going to help you remember that. In the endoplasmic reticulum, you see N-glycosylation. Actually, there's a special molecule called dolicol phosphate. Dolicol phosphate is a molecule derived from cholesterol, and you can start building sugars en bloc onto the dolicol molecule, and then those sugars are transferred as a unit over to the amino acid. Which amino acid gets the sugars through an N linkage? It's asparagine. So one of the important things to remember about the amino acid asparagine is that that's what gets N-glycosylated when proteins get N-glycosylated. Now, from the endoplasmic reticulum, we're going to move into the Golgi. So, via vesicular transport, the proteins are going to be transported to the cis, the medial, and then finally the trans Golgi by little vesicles. Something I want to remind you is that within the Golgi, right? Golgi is where O-glycosylation happens. So proteins that get sugars added through a hydroxyl linkage, the hydroxyl group having probably been added back in the endoplasmic reticulum, well, those proteins are going to get their O-glycosylation happening within the confines of the Golgi. After the proteins leave the Golgi, They can go to intracellular organelles, such as the lysosome, such as the nucleus. Maybe they'll go back to the endoplasmic reticulum. If that's the case, there's specific sequences that help them do that. Or proteins can be secreted from the cell. Proteins could be embedded in the cell membrane. It just depends on the protein. One other modification that you might be tested on, very frequently people are tested on this, is a phosphorylation of mannose. There's an enzyme, it's called phosphotransferase. Phosphotransferase is an enzyme that will phosphorylate specific mannose residues that have been added to specific types of proteins. What specific types of proteins are these? These are lysosomal hydrolases. Now, what is a lysosomal hydrolase? A lysosomal hydrolase is an enzyme such as a protease that cleaves proteins, a glycosylase that cleaves sugars, or a lipase that cleaves fats. It's a special enzyme that's going to degrade fats, sugars, and proteins within the confines of the lysosome. So how do we get these enzymes that need to work in the lysosome? How do we get them to the lysosome? It's through this phosphorylation signal. Phosphorylation of mannose creates mannose 6-phosphate residues on the lysosomal hydrolases, which then can bind to mannose 6-phosphate receptors within the Golgi, and this binding event traffics these enzymes to the lysosomes. It's kind of like this. The lysosomal hydrolases are like the garbage workers of the cell, and the lysosome is the dump. It's where the proteins, fats, and sugars go to be degraded. So we have to get the garbage workers to the dump so they can do their job. That's the job. So there's a card has to be handed out to the enzymes to get them there, and that's the job of phosphotransferase, to get the hydrolases delivered to the lysosomes. So why are we spending so much time talking about this? It's because there's a disease involved, right? Anytime we're going to spend some time talking about things, it's because it's an area where they're going to test you on the exam. So the disease we're talking about specifically is eye cell disease. Now, eye cell disease is caused by a defect in the phosphotransferase enzyme. So proteins, as they get folded properly in the ER and moved over to the Golgi network, the ones that are destined to go to the lysosomes are going to get phosphorylated on their mannose residues. If the enzyme that's responsible for this phosphorylation event is missing, where are the enzymes going to go? They're not going to know that they need to go to the dump to do their garbage work. They're going to be secreted from the cell. So what ends up happening is all the food that we eat and gets ingested by our cells, the fats, the sugars, the proteins, well, they're going to get delivered to the lysosomes eventually, but the garbage workers are on strike. There's nobody there to degrade the fats and sugars and proteins. So these are going to build up and up and up, and they're going to form inclusion bodies within the cell. Now, so eye cell disease is also known as inclusion body disease. It's caused by a buildup of inclusions within cells. Why are they building up? It's because the enzymes that degrade our foodstuffs are not there. Now, the second half of the problem is that the enzymes, instead of going to lysosomes, are going to be secreted from the cell. And this is going to cause proteins, sugars, and fats to be degraded in our extracellular tissue. That's not a good thing, okay? So you're going to get inflammation in areas you shouldn't. So let's look now at the major symptoms of eye cell disease so that if presented one of these cases, you can nail it. Now, somebody with eye cell disease, they're going to have coarse facial features. Sometimes these are referred to as gargoyle faces. Gargoyles are the evil-looking spirits on the sides of buildings that are evil-looking creatures supposed to ward off the evil spirits. Gargoyle faces. There may be gingival hyperplasia and even macroglossia, enlargement of the tongue. Developmentally, you're going to see craniofacial abnormalities, joint immobilities, clubfoots, maybe even a claw hand. You're going to get problems with the nerves that innervate the hands. You might even get scoliosis. These patients are typically going to present with psychomotor retardation, growth problems, and unfortunately, they're usually going to succumb to cardiorespiratory failure. These inclusions are going to cause problems in their heart. Their heart's going to be the organ that suffers, and they're going to die from cardiorespiratory failure, usually within the first decade of life. So a very devastating disease. It's one of the diseases that you might be asked about with regard to gene therapy. Now, theoretically, you could treat eye cell disease by putting in a copy of a gene that encodes phosphotransferase. If you could get phosphotransferase expressed in all the tissues of a person's body that normally lack the enzyme, well, then they might be able to live a normal life. Hasn't worked out perfectly, but it's a question that you might be asked on the exam.